Good morning, everyone. My name is Alex Harris, and I'm one of the Grand Rounds co-directors, along with Drs. Aditya Ladia and Rebecca Mule. Welcome to the first Grand Rounds of the year. Next week's Grand Rounds will be given by Dr. Reuben Robbins, Associate Professor in our department. The title of his talk will be Increasing Accessibility, Accuracy, and Fairness of Neurocognitive Testing for Diverse Global Populations. Okay, quick question for the faculty in the audience. How many of you know that you can request a meeting with the Grand Round speakers? All right. I see about 4 or 5 hands. If you want to contact us to meet with one of the upcoming speakers or to suggest a speaker, please write to us at grandrounds@nispe.columbia.edu. As a reminder, if you're participating today via Zoom, you may post a question at any time during the talk. Please use the Q&A feature, not the chat feature. As the online moderator, Dr. Rebecca Mule will forward the questions to me for our speaker to address. We encourage trainees to consider asking a question, and we will have time for trainee questions in person and online. Please indicate in the Q&A if you are a trainee. It's now my great pleasure to introduce today's speaker, Dr. Suzanne Amari. Suzanne earned her MD and PhD in molecular and cellular physiology from Stanford University, where she conducted research under Stephen Smith using microscopy techniques to study synapse development. In 2013, she established the Translational OCD Laboratory at the University of Pittsburgh, where she this is not the introduction that I wrote, but it's okay. We're going to go with it. Where she leads an interdisciplinary research team to develop better treatments for OCD. When I was looking over her CV, one of the things that struck me is that she did her, she came here to do her psychiatry residency and then did a T32 training with both Renee Henn and Blair Simpson. And one of the things that struck me while looking through her publication list is that you really see the influence of both of those people in like really high impact papers on clinical research into OCD, like TMS, as well as microcircuitry of grooming behavior in mice. So really impressive combination. She also maintains a clinical practice and serves as the program director at Wellcome Leap, where she developed and directs the $50 million multi-channel psych program, which aims to create innovative treatments for anhedonic depression. Suzanne's research has received support from the NIMH, the NINDS, and foundation grants, including a Burroughs Wellcome Career Award, One Mind Rising Star Award, several other foundations. Also received a number of awards in recognition of her translational neuroscience efforts, including a McKnight Scholars Award, an NIMH Brains Awards, the A.E. Bennett Research Award, the MQ Fellows Award, and the ACNP Eva King Killam Research Award. She sits on scientific advisory boards of several research foundations, including the Brain and Behavior Research Foundation, ORCHD, the International OCD Foundation, and ARIA, and is a council member of ACNP. But perhaps what she's most famous for on the internet are the heartfelt science ditties that she sings to the tune of Disney musicals. Check them out. The title of her talk today is Windows into OCD, Translational Insights from Humans and Mice. Please join me in giving her a warm welcome.
Thank you so much, Alex, for that introduction and for the invite. And okay, so someone is magically changing this so that my talk comes up without my doing it. Excellent. So it is such a pleasure to be back here. And I just looking out on the audience and seeing so many colleagues, friends, mentors. It's really heartwarming to be here and exciting and also a little bit surreal. I think the last, I saw Deborah Cavanis right before this, and I think the last time I was on this stage was when I was presenting my chief resident grand rounds in 2007. And it was about empathy in relationship to a long-term therapy case that I had done. And so this is going to be a very different talk today. And a lot has happened since then, but it's really fantastic to be here. I just want to advance through my disclosures, and I have no commercial interest to disclose that are relevant to the talk today. So since 2013, I've been at University of Pittsburgh, And my research program at University of Pittsburgh focuses on trying to understand the molecular, cellular, and circuit mechanisms that underlie abnormal repetitive thoughts and behaviors. And as you can see from this Venn diagram, these are common behaviors across a variety of different neuropsychiatric illnesses, ranging all the way from repetitive muscle contractions, such as you might see in something like dystonia or spasticity, all the way up to more repetitive, maladaptive, complex actions, such as you might see in something like addiction. And although I've labeled this here on this side of the axis as more complex or volitional, I put the volitional in quotes because it's really debatable how much control people have over actually doing these behaviors. But in particular, my clinical and research interests are really focused in on trying to understand the neural substrates underlying the obsessions and the compulsions that are central to OCD. Now, in this audience, because you have Blair Simpson and the wonderful OCD and anxiety clinic here, this OCD probably needs very little introduction. But nevertheless, I want everyone to be on the same page when we're talking about this. So according to our current DSM-5 criteria, OCD consists of either obsessions or compulsions, obsessions being repetitive, intrusive thoughts, that are either thoughts, impulses, or images, and compulsions being repetitive behaviors or mental rituals that people do that tend to neutralize the anxiety associated with those intrusive thoughts. Now, even though you don't have to have both obsessions and compulsions in order to fit criteria for OCD, most people do. And I think that's interesting in that it might tell us something about the neurobiology between these particular symptom constructs. OCD is extremely common, affecting between 1.5 to 3% of people worldwide. And despite the often flippant way that it's treated in the media, it can be incredibly disabling. But despite the fact that it is incredibly severe and highly prevalent, we still have relatively limited treatments for OCD, particularly when it comes to pharmacotherapeutic treatments. So serotonin reuptake inhibitors, or SRIs, are the only proven monotherapy for OCD, but they only lead to full remission in approximately 10 to 15% of people. And while exposure therapy with response prevention can be highly effective, it can be quite challenging to find skilled, trained providers to deliver it. And even if you have one, it can be very difficult to maintain the treatment and complete it if you're the person undergoing it. And so the bottom line is I see OCD as an illness of opportunity, which may seem strange given how severe and prevalent, et cetera, it is. But here we have an illness where we have, compared to other neuropsychiatric illnesses, relatively well-defined symptom criteria that are pretty circumscribed. And we have a huge unmet need in terms of delivering better treatments to a lot of people around the world. And so within my lab at University of Pittsburgh, we really try to take a truly translational approach that I learned from the excellent training that I had here in order to try to develop new and better and improved treatments for people who have OCD. And so what I want to start with to give you a sense of what is the approach and the framework that we try and use in my laboratory to do this, I want to use a clinical example. So most people in the audience are probably familiar with contamination-related OCD. And if someone walked into the auditorium here and they had contamination-related OCD and they saw that I had a big bleeding cut on my finger, that might immediately trigger in their mind an obsession. I might get HIV from the speaker. Right? Now, that's the obsession in this case, and that's triggering what I'm labeling here as inappropriate fear and distress and anxiety. Inappropriate because often, if you interrogate the person who has the OCD when they're not in the throes of the episode, they can say, well, logically, I know that's not going to happen. Logically, that can't be, an airborne illness couldn't be transmitted through the room to the back of the audience. But nevertheless, in at least one model of how the illness works, that distress and anxiety drives rituals to avoid or decrease the anxiety, which are compulsions. And in this particular example, the ritualized compulsion would be hand washing to the point of developing lesions on the hands. Now, one of the really nefarious things about this illness, and that makes it so difficult to treat and so difficult for the person, is that that compulsive behavior can actually sometimes serve its intended and goal-directed purpose, which is getting temporary relief from the distress and anxiety. So in that moment of the behavior, they may feel better, but the problem is it is typically very fleeting. And instead, you have repeated emergence of the obsessive thoughts, repeated increases in the distress and anxiety, and continuous driving of these rituals to void or decrease anxiety until the person can exit from this loop. Right? Now, so when I take a step back and look at these symptoms in the context of the illness, but as a neuroscientist as well as a psychiatrist, I can think about this as a behavioral circuit, right? These are behaviors that are linked together in a circuit mechanism, not proved at all of the arrows and the directions, but this is our current hypothesis and working model of how this works. And now when I think about how I want to bring this into the lab, what we can actually do, because again, we can't model obsessions in a mouse, right? We can potentially model compulsive behaviors in a mouse. I want to take these elements of these symptoms and break them down into component processes that can be modeled in mice and then mapped into neural circuits. And so we have action selection in the face of distress and anxiety. We have action completion once you've gotten into one of these ritualized behavior loops. And one element that we're very interested in the lab, I'm not going to be talking about today, but we have lots of exciting emerging work. Is this process of negative reinforcement, which is when you remove the negative stimulus, that's actually reinforcing for the behavior to continue. And what are the processes that are involved in that? And of course, because I am a clinician and I always want to think about with these animal studies, how can we actually uncover things that are relevant to human treatment? Each of these points of these component processes can actually be used as an entry point for treatment. So if we can block compulsive behavior initiation, if we can slow down or block that process of negative reinforcement, or one real problem is that people, when you're doing exposure therapy with people, a lot of the times they find themselves in one of these obsessive compulsive loops, even though they're trying really hard to stop themselves from getting into it. And there's a lot of shame associated with that. But if you get into one, can we facilitate action completion so we can get you out of those loops more quickly? All of these are potential places to start. Now, if we're thinking about how to model these kinds of things in mice, the good news in the field of OCD is on the circuit level, because based on the past 15 to 20 years of research in the field from clinical studies, such as those done by Blair and colleagues, we have quite good evidence of at least which lampposts we should start looking under when it comes to the neural circuitry underlying OCD, based on these highly convergent human imaging studies, which continue to highlight abnormal function and connectivity between cortical, striatal, thalamocortical circuits, and specifically calling out orbital frontal cortex and anterior cingulate cortex as areas of the brain that have dysregulation, often hyperactivity in the context of OCD symptomatology. And if you are fortunate enough to get a treatment that's effective for you, be that an SSRI, deep brain stimulation, or exposure therapy with response prevention, the abnormal activity in those neural circuits tends to be reduced. Okay. And so this is highly suggestive that there is a connection between abnormal activity in these circuits and OCD symptoms. But of course, in humans, you can't test causality, which is why we want to use animal model systems and then also bring the animal model results back into the lab. So circuit modeling in mice can be very helpful for modeling OCD relevant behaviors. Now, I had the good news here. Of course, the bad news is coming. So the bad news in terms of OCD, if you are an animal modeler, has been genes, in the sense that twin and family studies have revealed a significant genetic component to the etiology of OCD, and the heritability is quite high, on the order of 40 to 60%. But in terms of currently published GWAS studies, when we look at OCD and the results from the IOCDF Genetics Collaborative, we so far have had 0 statistically significant risk genes in that GWAS study. But there's a heavy caveat here, okay? And I want to put the numbers here. That study, which is now outdated because there are new studies that are emerging, and currently we have some on medRxiv from a group that's related to the Nordic genetics initiatives. This study only had 1,465 cases and over 5,500 controls. Now we look at schizophrenia, where we have had great progress in terms of finding GWAS hits, and we have 108 significant risk genes. That required almost 37,000 cases and 113,000 controls. So I would highlight again, OCD has been understudied, underfunded compared to other illnesses, and I have data to show that if anybody's interested. But I think there's a lot of promise that's still here, and ongoing studies are increasing GWAS power and also trying to take a different approach, which is, I think, going to be particularly useful for animal models, which is trying to identify rare OCD variants. And there's actually things that are emerging in that area that will be published, I think, in the next year. So that's highly promising as a researcher. Now, I'm going to have a little bit of a pause here and then restart the science portion. But since I have this platform, I want to take that opportunity to highlight something that I think should increasingly be part of collective awareness in our field in general. And that is the fact that we should all know and should not acknowledge that the way that we practice science and medicine in our country and basically around the world is impacted by systemic racism that we have within our country and within our scientific practice. So in the spirit of working towards being more inclusive as a field, I want to share this Neuron article, which at this point is now a little bit dated in the sense that it was put out in 2020, but still I think it's quite foundational and important, and it's open access, and I encourage everyone to read it. And what it highlighted is an important new initiative that was started with Danny Weinberger, Kapsaraza, and Lecia Crumpton-Young as being some of the people who were driving it to ensure that people of African ancestry are included in brain research. So lots of examples are included in this article, but the one I really want to stress because of the genetics component that I was just talking about is that people, and I hadn't realized how significant it was till I read this article, that people of African ancestry had been up to that point in time systemically excluded from GWAS study research. And the authors made it very clear that this was not malicious, this was not intentional to exclude people. It was based on statistical decisions that were made in many different rooms along the way, because of the fact that the genetic architecture of the African ancestry is more complex due to migration patterns that happened. But the problem is, because these choices were made along the way by many different people, no one quite connected the dots that the inevitable attempt to increase statistical power by limiting the complexity of the work would ultimately lead to systematic exclusion of non-European ancestry people. So in that schizophrenia GWAS, there were no people of African ancestry in that. And in the OCD, it's not surprising, but there were no people of African ancestry in that. Now, this is changing. And I am not a geneticist, and so I don't have to deal with the challenge that comes with working with the complexity that's associated with that genetic architecture. But I just want to highlight it because I think we as I consider myself a real translational researcher, but also very heavily rooted in the clinic. And I think it's important for preclinical researchers to also be thinking about this, because if the genes that we find and are using to model the illness in animals are really only rooted in people of European ancestry, then the hope that we have of developing precision medicines based on molecular targets that we're identifying preclinically and taking into the clinic may negatively impact people who are already underserved by our current mental health care system. So it's something that I think we should all try and keep in mind when we can. So what I'm going to tell you about today in terms of the research program within my own lab is using this image that I actually took in my office up in Colban X7. The backdrop is a skateboard from Colin O'Carroll. Some of you may remember him in the audience, but I thought it looked cool. And we've got Freud and the rat from Ratatouille. And because I really do want to try and highlight how in the lab we are trying to take things from mice to humans and back again. And so I'm going to start with a human story about molecules, an animal story with cells, and then a multifunctional circuit story where we're going back and forth with humans and animals. And so first, we're going to talk about molecular changes in post-mortem human brain tissue samples and how they might impact brain network communication. Then go to animals. What are the network activity patterns underlying initiation and termination of OCD relevant behaviors? And finally, an example of translating findings from mice into people, because again, that's the overall goal, to try to get new treatments to people to help them. So now, in the absence of the genetic hits was when I started my lab. And yet I knew that I wanted to ground my lab in things that were relevant to humans. And so what I realized when I went to University of Pittsburgh is that we could start to look at that intersection between circuits and molecules by identifying transcriptional changes within regions that have been highlighted in OCD circuit-based studies if we could take advantage of the resources of the University of Pittsburgh Brain Bank. And so I had two intrepid students, Sean Pantadosi, who was a grad student at the time. Now I was happy to find out just before the break, got his K99 NOGA. So he's going to be on the job market in 2 years. He's amazing. And Britt Chamberlain, then an undergrad, now an MD PhD. She is sadly and happily graduating this year. And she's going into psychiatry residency. And if you can get her, she's fantastic. So I just want to highlight the two of them. And they combed the brain bank, and which has been really geared towards illnesses that have not been OCD. They only found 8 people with OCD and 8 comparison subjects who had agreed to donate brains in their lifetime after their death. And so we decided we didn't think we were going to get much with that low of numbers, but we decided to go for it. And what we did was looked at 2 orbital frontal regions, caudate and nucleus accumbens, and did fresh frozen tissue, both qPCR in a targeted way, followed by RNA-seq. And so we were surprised. I really thought, given potential heterogeneity, we weren't going to see anything with those few of subjects. But what we found is that when we looked in a targeted way using qPCR at what we're calling here a synaptic composite score, so this is just grouping all of the glutamatergic synapse genes that we looked at together, so it's easier to look at and you don't have to look at 20 plots. We saw a significant downregulation in expression in the orbital frontal cortex, and here a trend towards downregulation in the caudate and the nucleus accumbens as well. And we saw a similar pattern when we look at glutamatergic receptors and transporters. For sake of time, I'm not showing you that in the inhibitory side of things, we did not see anything significant. So it really pointed us towards the glutamate system in these particular regions in the brain of people with OCD. But we wanted to take a more unbiased approach as well. And for this, partnered with outstanding collaborator Bernie Devlin at University of Pittsburgh and his colleagues Laura McLean and Bert Kley, and looked at differential expression in the 3 regions treated jointly, OFC, caudate, and nucleus accumbens, as well as caudate and nucleus accumbens on their own. And in general, we found a total of 904 total differentially expressed genes, with about half of them upregulated and about half of them downregulated. So that in and of itself doesn't tell us that much. But then what we wanted to do is go on and look at what potential gene networks were important within this upregulation and downregulation of genes. And the clusters that we saw, I'm not going to get into the details over here, but the clusters that we saw were really interesting. We saw 4 different clusters. And the primary one, the synaptic signaling cluster, contains 16 of the Go gene sets that were of interest, and including the glutamatergic system gene set that's relevant to that targeted qPCR. And consistent with our qPCR findings, we saw a downregulation in the gene expression in those regions. And then the other primary cluster that we saw was one that was taking us in a place that we hadn't expected. This was tyrosine kinase and circulatory system development, like a real vascular cluster. And here we saw an upregulation. So that was interesting. Now, we wanted to take that even further. And so we partnered with Jiebo Wang, who uses deconvolution approaches, also at University of Pittsburgh, in order to try to identify what cell types those different changes are happening in. And what he found was that we had decreased expression of these elements within striatal neuron cell type fractions. And what this means, we're calling them cell type fractions that means either that you have decreases in the numbers of those cells or you have decreased expression in those cells. But this particular analysis cannot dissociate those two. So it could be that there's decreased spiny projection neurons or medium spiny neurons in OCD, or just decreased expression of these proteins there. And then we also had an increased astrocyte cell type fraction. And this was when we were looking at all genes, all regions together. But then and in addition to this, we had an increased vascular cell type fraction. So increased vascularization expression pattern. And then when we separate out into the different regions within the orbital frontal cortex, we saw a significant, though relatively small effect size decrease in interneurons and an increase again in vascularization. And in the caudate, not surprisingly, that's where we see the spiny projection neurons coming out. So if we try to put all of this together and create a model, what we can then have, if we now have here thalamus, orbital frontal cortex, and striatum, we have evidence of either decreased PV neuron presence and or decreased function of those PV neurons within the orbital frontal cortex, which could potentially relate to these findings that we see in the clinic of OFC hyperactivity. We also have evidence of increased vascularization, which could be cause, could be effect of that OFC hyperactivity. And we have evidence of decreased spiny projection neurons within the striatum. And so that led me to have a lot of different ideas, including one idea, because we have overall down regulation of glutamatergic synaptic transcripts within the orbital frontal cortex. It really made me think more about the thalamus and how potentially the thalamus, which also is shown to have hyperactivity within OCD human imaging studies, maybe because there's so much overdrive to from the thalamus to the cortex kind of has to turn down that signal. And so we looked at that, and the idea that findings may suggest upstream thalamic pathology with the International OCD Foundation and Shale Springer, who's an amazing graduate student in the lab. And what they found was that when they did exquisite dissection of all of these different thalamic regions and did a ton of effort to find, to do these RNA-seq studies, that there was absolutely nothing that they saw on RNA-seq, which for trainees in the audience is still a really useful finding, because what that shows us is that we can convincingly say, at least on the gene expression level, that where it's at is in the orbital frontal cortex to striatum. But Shale did not give up. They persevered and also wanted to look at the proteome, because just because there aren't changes in the transcripts doesn't mean there aren't changes in the proteome. And these are hot off the presses data, and so I don't have slides to actually show it. But in the proteomics, we're actually seeing some really interesting stuff, specifically related to differential expression of inflammatory response proteins, and so upregulation of platelets and inflammation. So we're diving into that in more detail, and we'll be interested to see how that pans out. So this is giving us some ideas about the potential molecular architecture of the brain of someone who, at least in their lifetime, it's postmortem tissue, which has caveats associated with it. But the other thing I wanted to note before moving on to the mouse stuff is, even though it is a small sample size, it's actually, the findings have been overall independently replicated now by another similar small cohort from Brazil. So we're at least seeing some convergence amongst these findings, and we took their data set as well and folded it into part of one of our recent papers to again directly compare the findings. And given heterogeneity and all of those patients, they weren't all lockstep, the same in terms of comorbidities, medications, et cetera. It gives us some inkling that we may be onto something here in terms of the right track. So now I'm going to move on to how do we link all of those genes, and how do we think about the impact of these genes on neural circuits that are related to OCD. And the part one of this part of the talk is going to be focusing in on action selection and compulsive behavior initiation. And the second part with action completion, with the specific questions to be addressed in part one, is how are compulsive behaviors initiated in the brain, in this case of mice? And in part two, how does the brain know when an action is complete? So really trying to bookend these two things. How do I start? How do I stop? When do I do it? And in particular, to kind of decrease the state space, what we're focusing in on for this part of the studies that we're doing is looking at this phenomenon of striatal hyperactivity in OCD and trying to take it from humans into animal studies. With two specific questions: how is striatal dysfunction related to compulsive behavior? And can striatal dysfunction cause compulsive behavior? So for this particular section of the talk, in order to model compulsive behavior and striatal dysfunction, we're using a knockout mouse called the SAPAP3 knockout mouse. It was initially developed by Guoping Feng back in 2007. And when Guoping's group knocked out the SAPAP3 protein, what they found was that the animals engaged in compulsive grooming. And I know compulsive is a loaded term, but I'm sticking my neck out and I'm saying it because they keep grooming to the extent of developing lesions on themselves, despite the fact that they have intact pain sensation. It impairs their ability to eat, drink, sleep, care for their pups, etc. And yet they persist in doing it. So it persists despite negative consequences. And also in Guoping's initial work, they found that if they rescued SAPAP3 within a part of the striatum spanning a part called the central striatum, they saw complete resolution of this behavior. So putting it back in led to resolution of the behavior. Now, I want to be really clear. The SAPAP3 knockout model is not a mouse model of OCD. These mice do not have OCD. And I would have said that even if Josh wasn't in the audience. I really, truly, firmly believe this. I think it is a model that is very useful for the study of compulsive behavior and generates lots and lots of trials of compulsive behavior so that we can map it onto neural circuits using the techniques that we've developed to do that. But I am a little bit more optimistic, at least, that the findings that we find in SAPAPs may be relevant to the disorder in some way, at least in some subset of people, based on our human postmortem work. So we see both in the orbital frontal cortex and in the striatum, there is overall a downregulation of, it's called DLGAP3 in humans, but the SAPAP3 homolog. And therefore, it's at least going in the right direction in terms of the protein that is involved in the model system that we're using. So now we see Sean Pantadosi again, who not only doing the human postmortem work, also his primary project was doing the animal work. And what he decided to do was look in the striatum of these animals using InVista microscopes from Inscopics. These are microscopes that the mouse can port around on its head. And so I'm giving you an example movie of this happening right now. And so you can see the mouse is freely moving around and can engage in grooming behaviors such as seen here. And then you're looking down the lens of the microscope, and each of those white flashes of light is a calcium signal. So you're seeing a little network of neurons within that 500 micron diameter of the lens, and then we can map those neural traces onto the behaviors that the animals are doing. So we inject the virus, wait 5 to 6 weeks after implanting the lens, align the base plate and focus. And for everything I'm going to be talking about today, these are bottom-up views so that we can really resolve and find resolution, the grooming behavior of the mouse, and then we can map it to particular cells as seen in this particular image here. And so the first thing it is crucially important to define your behavior before you start to engage in any kinds of these monitoring of neural activity things. And so we first wanted to really define the grooming behavior. Not surprisingly, in our hands, SAPAP3 knockouts had excessive grooming at baseline. They had more grooming bouts in this particular set of cohorts. But we were also interested in the pattern and the quality of the grooming. And so this is a standard mouse grooming sequence. For those of you who are not familiar with it, they go kind of top to tail. And then if you look at the SAPAPs, though, and have not seen thousands of hours of grooming videos like I have, this is different because they engage in facial grooming, and then you'll see that they go to a scratching element, facial grooming and then scratching. And then what it almost seems like when you watch enough of this is that there's actually a stop here before they go on to proceed down all the way to licking the flank. And another fantastic graduate student, Victoria Corbett, she actually quantified this by separating the knockouts with high grooming, low grooming, and wild types into different bouts to try and determine if they were like more organized or sequential versus less. And what she found is that when you look at something that we're calling the transitions to bouts ratio, meaning like how many times do you go back and forth before you actually complete a bout, the knockouts have a lot more of that than the wild types do. It's higher for them. And so that's showing us they have this kind of interrupted cyclical pattern that they can't get out of. Now, the first thing Sean did was look in the central striatum, that area that we think is important. And I have these lollipop diagrams up here to show you which cell types we're looking at. This is a combination of both D1 and D2 cells for those in the audience who care about that. And so when we just look at bulk signals of all of the striatal neurons together, we see an overall hyperactivity in the knockouts compared to the wild types. Only when they're grooming, though. When they're not grooming, we don't see a significant difference between these. But this really doesn't capture the use of the calcium imaging technology. So what we really want to look at is the time resolved neural activity, with time here in relationship to the grooming events being on the x-axis, and time 0 is when the grooming started. And here we have wild types and knockouts, and this is a heat plot of all of the different cells lined up on the y-axis. And so if you take a step back and look, you kind of get the impression that there's warmer colors here in the knockouts, if the contrast is good on the screen, and the wild types. But to visualize it more easily, if we average the whole, all of the cells together, you can see a dramatic increase in the Z-scored fluorescence, so in the calcium activity of the knockouts compared to the wild types. Both of them have an increase at the time of grooming, but it's much more significant in the knockouts. Right. So I want to pause here because we were very, very excited about this result. And so I want you to understand that excitement, why it was so exciting. And it was because for a while I had been searching in a holy grail kind of way for anything in the brain that we could hang our hat on as a signature of a compulsive behavior. And why do I care about that? I care about that because if we have compulsive behavior signature in the brain and we can find it in animals and then we can try and identify it in people, then that sets you up for potential closed loop deep brain stimulation that you can do in humans if you can sense the signature and then deliver a stimulus that's appropriate to it. And here what we have is two sets of animals engaging in completely the same behavior. Like if you look at the grooming, it looks like grooming. It's not as if it doesn't look like normal grooming, just like someone with OCD who's washing their hands. They're not doing excessive abnormal movements when they're washing their hands. They're washing their hands. It's just that it's cyclical and it's in this repeated pattern. So phenotypically, we have the same outwardly demonstrated behavior, but internally, the motivation is different in some way. And this is the first time we were able to see that, to see this kind of signature. So then we wanted to dissect it further. And looking at the cells that are positively or negatively regulated in response to the behavior, we see that more of these neurons are active at the onset of compulsive behavior, which is consistent with what we've seen. And then we really wanted to dissect out which cell types were responsible for that. And there have been a lot of theories in the OCD field about either excessive activity in the direct pathway or decreased activity in the indirect pathway, potentially promoting OCD. So we thought, well, here's the perfect system. We can directly look at this and see what D1 or D2 neurons are doing in response, with our prediction being that D1 neurons would be hyperactive at grooming onset. Now, so we did some crosses of the mice to the D1 Cree mice so that we could just get expression of that calcium indicator in D1 animals, and saw exactly the opposite of what we were predicting to happen. So here the knockouts have decreased grooming-associated calcium event rates. And when we look at the time-resolved average neural activity, you see that there's effectively very little difference between knockouts and wild types, with a tiny bit of a bump after, a tiny but significant bump after grooming onset, but in a delayed way. So this had us really scratching our heads. We now have this model of D1s being lower, and we didn't know what D2s looked like. but we do know what D1 plus D2 looks like, and we can do arithmetic. And so we proposed a year before we actually got the data that the D2 neurons must be doing something. And so this was an experiment that had us really holding our breath, and we didn't know what we were going to see, but we had this prediction, and it was one time when it actually came according to what we were predicting, with there being a very significant increase in D2 activity that was happening at the onset of the grooming events. So this didn't explain things any more to us, though, than what it gave us even more questions about how might elevated D2 activity lead to abnormal behavior. But then we went back to the literature and found that Patrick Rothwell, when he was still in Rob Malenka's lab, had done some really interesting work on a task where you have to have a mouse press lever A and lever B, and then it gets a reward, and found that they had increased D2 SPNs when they perseverate on a lever and don't do the right choice. And then another person, this was Shinjin's lab at the Salk, had a similar task, but 2 left sequences, 2 right sequences. And also they had increased D2 spiny projection neuron activity when they were switching between these different subsequences. And so you get some concept that elevated D2 activity may be important for perseveration and switching and similar things as to what we see in the SAPAP knockout mice. So again, in mice, we can start to test causality of these things. And so what we did was inhibited D2 SPNs. Specifically, we inhibited, for aficionados in the audience, we inhibited indirect pathway SPNs because we had to use a retrograde approach to go into the GPE and then get back to the D2 SPN, to the neurons in the central striatum. And when we did that inhibition over 20 trials, what you can see here in the control compared to the inhibition with ARCH-T, the black stripes are grooming events. And you can see there is, in this one example case, you see less grooming during the ARCH inhibition. And this is the overall decreases that you see in the ARCH condition versus the knockout condition. So we have evidence that inhibiting D2 SPNs does decrease grooming probability in SAPAP3 knockouts. Okay, but I told you I was going to tell you how our compulsive behavior is initiated in the brain. This doesn't get us there. And so one question that then Victoria asked in my lab was, is direct activation of this part of the striatum, is that sufficient to cause grooming? And so she put channel rhodopsin, both sides, into the striatum and then pulsed the laser on. And what you can see if you look closely is when the laser comes on, the animal starts to engage in little snippets of behaviors that look like grooming, but it's not full grooming episodes. So laser on, it kind of turns and then it stops. And what was interesting is these little snippets of grooming behavior, they actually came on at the time of laser onset, but then decreased before the laser was complete. So that was interesting. And it did look like initiation of this grooming behavior in wild type mice. So what we have now is evidence that the central striatum is activated at compulsive grooming onset, that D2 neurons in the striatum are responsible for that activation, that direct activation of this part of the striatum leads to fragmented grooming. And I didn't show you this for sake of time, but increased activity in the striatum is specific to grooming and not to running around locomoting, not to rearing, not to other kinds of behaviors. So that left us wondering, okay, is there some link between the cortex and the striatum that might help us to get from this fragmented grooming to full grooming events? And what are the potential cortical drivers of this? And Victoria did beautiful anatomical work to trace the connections to the central striatum out, because it really has been very minimally looked at until the work that we did. And we were not surprised to see projections from the lateral orbital frontal cortex. That was what we were expecting. But funnily, we were surprised that it wasn't the majority, and that the majority was coming from this area that we're calling M2 here, but it's called a lot of different things. You may be more familiar with it as being ALM. And so we're going to call it either M2 or ALM, anterior lateral motor area here. And so that was interesting because then when we looked at knockouts at these projections from M2, we saw compared to wild types, you had a significant increase in EPSPs in the knockouts compared to the wild types, with the max EPSC amplitude being significantly more in the knockouts. So this is a stronger projection despite no differences in intrinsic excitability. And then if we go and do the experiment where we activate these projections specifically, the ones going from M2 to central striatum by injecting up here in M2/ALM and then putting our probe within the central striatum, what you then start to see is when the laser comes on, you see the mouse start to engage in a full grooming bout, and it looks as if it is a normal grooming bout. So, and we have some that respond, some that don't respond, and the non-responders, essentially there's a non-significant but a correlation between how much virus they have and how much of the area is being activated. Now, what's again a little bit weird is this time course. You see this delay here, that it doesn't start right away, and it emerges and then peaks and goes off as soon as the laser turns off. And we were interested and curious about that because it wasn't what we were expected. So we simultaneously put in an opsin into the ALM and then simultaneously stimulated and imaged the cells within the striatum so that we could see what was happening when we did that stimulation. And interestingly enough, there is also a delay in activation of those neurons after you do that stimulation. So essentially the behavior is mapping onto the time frame of the activation patterns. So this is evidence, again, that we have now saying artificial activation of M2 and CS leads to full grooming events. So this starts to give us this model that potentially central striatum might even, in other experiments I didn't show you for sake of time, it looks like striatum is leading ALM if you're actually looking at naturalistic behavior. So possibly it kicks things off and then ALM comes in. Now, but part two, okay? I promised you to talk about what I actually think is a fascinating question. How does the brain know when an action is complete? And I don't mean like the traditional things similar to what I did in Blair's group, looking at stop signal task, right? That's a cued action. You know you're supposed to stop. I'm talking about things like brushing your teeth or washing your hands. You stop at some point, but there's nothing telling you when to stop. There's no cue. So how do you know? And Blair will know why I care about this as an OCD clinician, is that feeling of completeness, that feeling of, oh, it's just not quite right, that feeling of I need to do it more, I need to do it again. How can we capture that? And so then we return to M2 ALM for this part of the story. Because what we found when Victoria looked at ALM, now we're using fibrophotometry, and looked at activity patterns in there, and in gray, you have an animal that's grooming. We kept seeing little peaks of activity when the animal was engaged in grooming behavior. And if we average it all together, you see a significant increase in activity in ALM now when grooming is occurring. But we don't see anything like this in either rearing or locomotion. Again, it seems very specific to this repetitive grooming behavior. And what we saw, Victoria used a function to try and find exactly when the peaks were happening, and then when grooming was stopping, and then mapped these out and found if we it's easier to look at if you separate it out by the length of the bouts. But we have 0 to 2 second bouts, 2 to 4 second bouts, 4 to 6, and 6 to 8. And you see that as grooming is occurring, we kind of have this increase in activity that peaks and then drops off, and does so no matter how long the grooming bout is. This is highly correlated. The length of the grooming bout is highly correlated with the M2 activity peak, again, suggesting potentially that there's something about this signal that it's definitely associated with when this action stops. Whether or not it causes the action to stop is another story entirely that we're starting to get at. This is our first effort at trying to get at this and looking at inhibition of central striatum cells that were receiving projections from M2 using a virus that we're trying different approaches now. I'll just leave it at that because there is some toxicity associated with this virus. But our early efforts into this showed that in some animals that had inhibition in the central striatum there, we saw a decrease in grooming in SAPAP knockouts compared to wild type animals. So we have ideas and theories that we're continuing to now pursue in NIH-funded research of how does the brain know when an action is complete. It seems that CS initiates these fragments, that M2 may sustain these complete action sequences, and M2 activity peaks at bout termination, with the remaining big picture question being, what is the terminator? Because we don't know, right? The termination of these sequences, it could be something where the GPE is coming into the striatum. It could be M2. It could be microcircuits within M2. This is currently being investigated, but we're really excited to try and figure this out because, again, you can imagine that if we can figure out what is helping people to understand when an action is complete, this may help us to define new therapies for people who have OCD. So the final section, and I promise it's a short one, but to me, it's the most important one because it's why we're doing this. We want to get better treatments into people. And those of you who were at Columbia at the time will remember the study that I did with Renee, Blair, Josh, and Tim Spellman, where we took optogenetics, which at the time was fancy and new and shiny, and we injected channel rhodopsin into the orbital frontal cortex of mice and then stimulated them at this orbital frontal cortex to striatal projection. And what we found was that if we did repeated corticostriatal stimulation at that particular synaptic pathway, we saw increased grooming not during the time of the stimulation, but after. So brief but repeated stimulation led to increases in grooming behavior that was very nicely correlated with increased evoked activity at that corticostriatal synapse. And that was beautiful work done by Tim and Josh's lab at the time. And not only that, but if we stopped doing any kind of stimulation, came back 2 weeks later and did a measurement of the grooming behavior at the time, it was still elevated, though not as significantly increased as it was at that time. So suggestive of the idea that by hyperstimulating this projection, that might lead to OCD relevant behaviors. Now, that is suggesting that there's plasticity that is happening, but for not good reasons, right? I mean, the reason is agnostic, but it is leading to not good things. But it raises the question, of course, is can plasticity be leveraged in a good way? Can we get in there? And we all know that deep brain stimulation for OCD is now approved under humanitarian device exemption from the FDA. But the problem, of course, is DBS is invasive and not everybody wants to have brain surgery. So what this leads us to think about, though, when we start thinking about plasticity being at these different nodes, is that if there is plasticity at this particular synapse, then potentially we can engage cortical circuits in a non-invasive way to affect plasticity downstream. So this was a question that we wanted to ask, and I partnered with Dr. Rebecca Price. Becca, many of you may know her. She's a fantastic psychologist and researcher at University of Pittsburgh, and I was fortunate to meet her very early on in my time there. And what she did, she was one of the pioneers of doing deep orbital frontal cortical TMS, along with Colleen Hamlin. And together we worked on this study where we had a hypothesis that if we downregulated that activity, consistent with our findings from the animal research, where upregulating was bad, downregulating would be good. So if we downregulated activity using continuous theta burst stimulation, that would help people with OCD. Comparing it to intermittent theta burst stimulation, which would either be neutral or hurt people with OCD. The idea was hopefully it would be neutral. And so Becca did this in combination with a particular therapeutic intervention, a habit reversal intervention. And then we have baseline here. And then in orange, we have the upregulation. In blue, we have the downregulation, and you see a single intervention of the continuous theta burst stimulation led to a decrease in urge strength, a decrease in time spent in compulsive behaviors, and a decrease in effort needed to resist compulsions. And so this was incredibly exciting to us because we could actually take the results from the animals and test the hypothesis in the humans, and it was consistent with what we'd found in the animals. And right now we're in year 5 of the sham compared to the CETB, compared to this downregulation intervention to see if it comes out in clinical trials. And so that is really exciting, and we're going to break the blind, and maybe it'll work, maybe it won't. Hopefully it will, because the goal of all of this is to develop improved neuroscience-based treatments for OCD, not to, I care deeply about basic science and discovery-based science, and it's critical for the work that we all do to continue to do that. But for me, what I gain the most pleasure in is trying to then bring these findings into people. With that, I'll end with a couple of things. Please consider brain donation. Without it, we can't do the kinds of postmortem work that we're talking about. And a lot of people don't realize that brain donation is different from organ donation. You have to do it separately. So consider doing that. We need all kinds of brains. And before I thank my lab, it's when you do postdoc and residency training somewhere, you don't get like the PhD thesis defense to thank people, but I have some thanks to give, and I wanted to put up a slide from like my very first Career Development Institute in Psychiatry talk about what I wanted to do in my postdoc and in my K award. And this was it in Comic Sans Serif and all. Our translational approach. I hope everyone agrees my slides have improved since then. But the idea is to have a great collaborative team. And I had one of Blair, Renee, and me. And to be I'm like, and I look a lot younger than it's been a while. But to try to really understand pathophysiology for not just drug development, but also circuit development. So thank you deeply for the training I've had here, for the mentorship I've had here, and continue to. And I want to thank my lab, my funding sources, and all of you for your attention. Thanks so much.